proteomics and Gagomics and Glycomics and Glycomics. And essentially, he is spearheading our entire effort with regard to the omics technology. And it's because of him that we have the phenomenal mass specs. I know some of you are already interested in his line of research. So I think it's good that he's here, and he's going to tell us a whole lot about mass specs, starting from the very, very fundamentals. Right?
Thank you, Rich. So the lecture series that I'll give today, the first half is going to focus more on technology, some of the fundamentals that will just help, I think, frame some of the discussions about applications and some of the things that make it kind of challenging to analyze GAGs and also in Glycans. And then on the back end of the presentation, we'll take a break, and then on the back side, we'll talk about some of the applications and a little bit of data, and hopefully get more dialogue going, because within about a 30, 45-minute discussion, it's a lot to kind of get too much, don't want to overwhelm you with too much data. So from the standpoint of like how many folks are familiar with mass spectrometry, you just get an idea, like when you taken like the medchem, have you ever taken anything that I've taught before, medchem? Yeah. So you guys will probably see some of the same slides, okay, because kind of overlap, but for the most part, I guess the first set will kind of focus on the technology. So again, the first part, overview of mass spec, will focus on ionization sources and some of the key mass analyzers. And then again, some of the common configurations for LCMS, and try to pull up some of or kind of push directions to the animation. So. Okay, so I like to start off most talks, beginning talks with mass spectrometry, to emphasize that mass spec is not a new technique. It's been around for quite some time. These are two of the founding fathers. J.J. Thomson discovered the electron, and his assistant, Francis Aston, discovered a lot of isotopes. He's also a very skilled instrument builder. And so between these two folks, this is really kind of the birth of mass spectrometry, and this really set in motion a lot of the techniques that we now have available to us. And the bottom figure that you see is one of his Aston's third generation mass spec. And there's some still key elements to this mass spec that are relevant today. And we'll talk about that on the next slide. The two main things that I want to focus, so what are some of the primary components of a mass spec? There are many. The two that we'll focus on, again, for today's lecture involve sample introduction or ionization sources, and then the mass spec component, mass analyzers. Okay? And there's lots of other components, of course, but those are the two main big ticket items. In terms of sample introduction, of course, in this day and age, you can introduce gases, liquids, solids. The solids can be introduced in the atmospheric pressure, which is kind of this orange box. And then you can also take solids like moldy, which we'll talk about here in a bit, into the vacuum system. So a lot of the tissue imaging that you'll see if we're doing PK/PD studies or looking at the tissue distribution of drugs gets done with moldy in the vacuum. I guess it goes without saying, but I say it anyway, you can't analyze anything unless it has a charge on it. You have to ionize it. Okay? And so in the early days, the first 70 years or give or take, you were really restricted to things that were, say less than 1,000 molecular weight. And to get those to ionize, you had to derivatize them and make them more volatile. And so really the first part of mass spec success, the first 75%, if you will, was focused on those low molecular weight species. And that's really what kept biological mass spec that we know today from becoming what it is. Then certainly with development ionization sources, electrospray and MALDI, which we'll talk about, really the ramp up in terms of mass analyzer development really kicked into gear. The companies got really involved. There was a lot more money involved, and so you start spending a lot more investment. So we now have just a huge, array, not a huge array, but I guess it's getting smaller and smaller, but the technology, the mass analyzer technology that we have is just profound in terms of its power. I'll talk about MS or MSMS or MS to the N. MS is generally just measuring the intact molecular weight of something, the intact form of a given molecule. Whereas MSMS usually means you're measuring the fragment of a parent or intact molecule. MS to the N can be 2 stages, 3 stages, and so forth and so on. Practically, you're really limited to MS to the 3rd or 4th, maybe. And then, of course, you're left with detecting either the intact form here at the detector or the fragments of the intact form. And critical to all of this is, going back to the original picture that I showed you, is that vacuum tube that Aston built. All of these things are in a vacuum system. So there's a big need to keep all of those gas molecules out of the way so you can get your charged molecules through all the different ion optics and to the detector. So this is just an idea of the early ionization, or an overview of ionization techniques, starting with the early ionization sources. The first being electron impact, which is just, that's still relevant today in GC mass spec. That's the primary mode of ionization. You're taking an electron, impacting a volatile molecule, and you create a lot of fragments. Okay? It's very harsh. So if we look at this electron ionization all the way through the fast atom bombardment, these are all very harsh things that are going to destroy any soft, malleable proteins or carbohydrates or anything like that. And so that's why, again, these early guys were restricted and just really couldn't do a whole lot with them. And then the contemporary mass spec, biological mass spec, really came into form in the late 1980s to early 90s, and that was really driven by the introduction of electrospray ionization and matrix-assisted laser desorption ionization. And those two things are considered soft. Okay, these things do not fragment these really large biomolecules that have no natural volatility, if you will. Having to get these things from a liquid phase or a tissue or a cell, to get it from that into the gas phase, you have to put charges on it, and you have to get into your mass spec. It's a pretty difficult transition to make. And so those are the two techniques that really kicked into gear. And these are the two gentlemen who really ushered that in, or the two main guys that got recognized for the Nobel Prize, Koichi Tanaka and John Finn, our own John Finn here at VCU, who's no longer with us, but of course that's when he won the Nobel Prize while he was in chemistry here. If you look at the timeline, I'm plotting here the number of mass spectrometry papers in PubMed. So if you just search mass spectrometry, plotted as a function of year, you can see that things were going okay in the 70s, 80s, and 90s, and then if you look at when electrospray was really introduced in 1984, when they were looking at synthetic polymers, but then in 1989 is when everybody's attention. It was like, whoa, they're ionizing intact proteins. And that was this science paper in 1989. MALDI has got a little different story, 1985, 1987, 88. Koichi Tanaka is not considered the guy who developed MALDI. He's just a guy who took a matrix, put a biomolecule in it, and got it to ionize. So that's why he got the Nobel Prize. But MALDI, I think 1985 is really kind of the pivot point there. And then, of course, there's this huge investment. Afterwards, you start seeing this huge surge in papers, and I think that you're starting to see this slight decline more recently, only because mass spectrometry is becoming so ubiquitous that you don't really find it a lot of times in the keyword or title. It's just kind of like, it's like UV-Vis or something like that. So anyways. So this is some of the development of MALDI. MALDI is not as commonly used in glycan analysis, although I'll talk about some examples. It's really got the market, or really what's used for in, say, the pharmaceutical industry, is for tissue analysis, as I mentioned. So if you're looking for where your small molecule is, liver tissue, you take a mouse or a rat, and you cryotome it all the way down, you can actually put an entire thin slice, 10 micron, whatever, 50 micron slice of a rat that's been dosed with your drug of interest, cover it with matrix, put it into the mass spec, and you can image it. Come back the next day and check it all out and see where everything is. The development of it actually started in the 60s, of course, where people were using laser desorption, trying to get things above 1,000, 2,000 molecular weight. And the real watershed moment was when Hillenkamp and Karas, who were really the two guys who were given credit for developing MALDI, started putting their analytes into organic matrices. And those organic matrices, which was, again, Analytical Chemistry 1985, that's kind of the paper I'm using as the reference point on the previous slide, is really where you started to get these ionizing small organic molecules and biomolecules less than 2 kD in these matrices. And again, as I said, Tanaka published this paper in RCMS in 1988, and then all of a sudden, everybody's like, well, oh my gosh, I never thought of just taking a protein and putting it in here and ionizing it, and all of a sudden, boom, it's a whole new ballgame after that. But again, Terrace and Hohenkamp did not get the Nobel Prize, so we're terribly happy. So what are some of the advantages to MALDI? It's amenable to offline collection liquid separations. So the early days of glycan analysis, you see a lot of these things with carbohydrates. Still soft enough for carbohydrate analysis, particularly in glycans, which is what you see a lot of because they're probably the easiest to extract a lot of it and get into a matrix form. There's an enzyme we'll talk about, PNJZF, other things. But it is harsh enough to where people worry about it because it can knock off sialic acids and other types of labile modifications. So some people probably have the expertise, the technical expertise and know-how to make it work and work well, but it is a technical issue that people do, it is a concern. A nice thing is it's less susceptible to ionization suppression, so we'll talk about electrospray, which really allows you to directly couple HPLC to your mass spec. In MALDI, you don't have to worry so much about, do I have 10 or 100 millimolar sodium chloride, okay? That is not as big of an issue in MALDI, all right? In electrospray, it's a big issue. Still soft, but again, like I said, there are some concerns about popping up some of the labile groups. Very high throughput, practical range. It does produce primarily singly charged species. And that is a problem, or can become a problem, when you're trying to do MSMS, or tandem mass spec. In shotgun proteomics work, where you have peptides or proteins, and you put at least 2, not 3 or more charges on it. Certainly at the peptide level, you've got 2 or 3 charges. Proteins, you just start dumping more and more charges on it. When you go to fragment that intact biomolecule, protein or peptide, you have multiple charges. It's got enough internal energy, and when I start fragmenting it, when it sends one fragment this way or that way, you've got a charge on it typically. You've got an N-terminus, and you have a C-terminus, which is a lot of times arginine, lysine, trypsin. So you've got these 2 positive charges. When you start whacking it in the middle with neutral molecules, you can analyze the different B and Y ions. Singly charged, you can only, you don't have as much internal energy in the molecule. It's just not as amenable to carrying charge when we fragment it, so it's not the best in that regard. This is what it generally looks like, whether it's an atmospheric pressure. Most MALDI is done in vacuum, although you can do it in atmospheric pressure. It's usually done with pulsed UV lasers, although more and more IR lasers are being used for different hybrid things like what's called Maldesi, which or laser desorption, followed by post-electrospray ionization. That's becoming more and more commonplace. Has a lot of advantages that we won't go into. But for the most part, UV was where everything got started because the absorbance of these organic matrices that you were using in MALDI had pretty high absorbance cross-sections with the UV pulse wavelength, okay? So I'll show you the next one, when you're firing that laser into this matrix sample plate, if you will, that UV light is being absorbed pretty efficiently with those organic molecules. IR, on the other hand, which if you know anything about IR, it typically gives you so it's usually in the 2900 to 3000, 3100 wave numbers. And that is the vibrational rotational frequency or absorbance bands for water, okay? So in a tissue or other types of things, IR is really desirable because if you freeze something in water, you've actually got a natural matrix around it. But the problem with IR, there's twofold problems. One is ionization efficiency in IR is always very low, very poor. Secondly, the cost of IR lasers used to be exorbitant. 10 or more times as expensive as a UV laser off the shelf. Okay, so most people are like, that's just too much. Now IR lasers are coming down in price, and ionization can be improved again with post-ionization techniques and other things. So it's maybe coming back. Who knows? We'll see. but also probably taps into maybe some other carbohydrate. Yes.
So, is it the charge on the ion that actually causes it to be like pulled from the plate, I guess, and travel into the mass spec?
Yes.
Okay.
And in MALDI, I've kind of whittled away a lot of the slides, so I'm not sure if I cover this, but the thing about MALDI, this is very different than electrospray, and I got my PhD in something called secondary ion mass spectrometry, which is a very niche, very niche area. It's very similar to MALDI in that the majority of things that come off the surface are neutral. Ionization efficiency is fairly low, all right? And in MALDI, there's something called the lucky survivor theory, where essentially you don't have, you have a, I would say, a net neutral charge, give or take, but as the plume comes away from the sample plate, you end up getting this, so the temperature of this laser when it goes into that matrix is like 700-800 Kelvin. So you get this thermal expansion, but then everything is starting to kind of, because you've also biased it, you've got this charge on it, and all of the matrix molecules, if you will, start to sublime. They kind of evaporate, if you will, off of the encased sample. And yes, as soon as you get to the grid, where you end up extracting and accelerating those ions to a kilovolt or more of energy, that is exactly right. You'll get this plume. The neutrals will just kind of go wherever they go, into the vacuum or on all the other coating area surrounding it, and then ideally the positively charged, primarily positively charged things are going to go through the grid. If you're doing it in negative ion mode, you can do that as well, but not a lot of people do negative ion mode. I know it's an acid, so you have to go to another matrix. Okay? So this just gives you the essential functions of the matrix. Analyte matrix, again, I kind of already mentioned this. The idea, the purpose of the matrix itself is to absorb the energy of the laser and lift it into the gas phase, ultimately ionize, extract by the optics, and analyze. These are some of the common organic ones. DHB is the most common, but there's lots of others as well. But DHB is the dominant one that I'm aware of. Also, synapenic acid and some other things. A lot of times it's just a trial and error to see which ones work. The conditions under which you make the matrix is the next one. This can have a huge impact. People have done crystallography studies of the actual matrix that you generate to see if it's do you get higher ionization in the regions that are highly crystalline versus the ones that are amorphous? Turns out from the studies that I'm aware of, or in my old groups, it's the more amorphous regions give you better signal than do the high crystalline environments. But I don't know how, I haven't used MALDI in at least 20 years. But in the old days, you used to have this little joystick, and you move the laser around like you're playing Atari. Like when I used to play, that's how old I am. But you're basically shooting the surface with this laser, and all of a sudden you're looking at an oscilloscope, and you're watching it, and then all of a sudden you'll see the signal go up, and you're just going save, and you're like, okay, I should graduate, save. So you're saving these spectra as fast as you can, but it's hard to find which ones are going to give you the hot spots or not. I've shown 2 different ways to make the matrix. There's a lot of art, the rate at which you evaporate, the kinds of solvents that you use, certainly the kind of matrix that you're using. All of these have a big role, and it's particularly the case for carbohydrates as well. This is just some data to give you an idea. Again, these are in glycans, okay, that have likely been released from some type of probably protein, plasma protein. So it's, I guess it's all of human plasma in glycans. So they probably took a blood sample, an EDTA, they spun it out, they pulled off the plasma. They went through a series of sample preps, which probably included reduction, alkylation, and digestion. And then they were left with your protein, your glycopeptides, and your regular peptides in this mixture. And then they treated it probably with a PNGase-F, okay, which we'll talk about in more detail later. And so what you're seeing are all these glycans that have been cleaved off of all of the plasma proteins and put into a matrix. This is, don't think it gives, I didn't look up which matrix they're using, but let's just assume it's DHB. And you can see that this whole series going all the way across the mass to charge x-axis is showing you singly charged in glycans with high mannose, hybrid amounts, some of the modifications like the salic acids, which I told you are, you can pop those off a little bit easier if you don't have the conditions right. Those are there. But as far as a quantitative readout, you got to be really careful with this. And electrospray is the same way, so it's not just a MALDI issue, but MALDI, you really don't want to look at this and go, oh, look how much more of this glycan is there versus that glycan. That can be a serious mistake. Okay? And so when we get into start talking about reducing in derivatization of N glycans, so if you can look at the cursor, and I'll show you, like right down here, okay, this is the reducing end of this glycan, the blue box. Okay, look now. That gives you a free reducing end, which you can then put a tag on, and that tag can, in our case, have a stable isotope labeled standard, so you can do quantitation. You might pool all of your patient sample blood together, run it through a process, and then derivatize it with a light tag. And then every other single patient sample, you put a heavy tag on. And so now you spike every patient sample with a heavy tag on it into your standard light standard. And so now you've got pairs that you can actually look at and get relative information. And it's not an absolute concentration, it's a relative concentration. So that's going to be something to keep in mind when we move to the second part of the lecture series, okay? All right, so electrospray, probably the more important of the two, so I don't want to probably spend too much time talking about MALDI, but that's all right. The big thing with electrospray is it allows for the direct coupling of LC, HPLC, or UPLC directly to the mass spec. That is a huge component that cannot be, you can't emphasize the importance of that enough in terms of this, people for years have wanted to couple, LC up to mass spec. They were stuck with UV vis, fluorescence, whatever the case may be, and they wanted that molecular specificity that mass spectrometry offers, but they were stuck because they didn't have a very effective way to ionize things. And so that's why electrospray really transformed not only mass spec, but also the whole biological mass spec as well. Multiple charging, that's a real important factor, which I probably won't get into as much as I could, but the more charges you put on a molecule, the lower the mass to charge is. So you measure in a mass spectrometer the mass to charge. So the more Z, or the charge, okay, the lower that mass to charge will be. So a lot of things get better with analytical figures of merit. Detection limits, resolving power, things of that sort. So multiple charges can really help you. It also helps you when it comes to fragmentation, like I've already mentioned. It is the softest ionization technique known. You can do structural analysis, practical mass range. There is no, I don't know what I would tell you as far as an upper limit of what you can analyze, because people continue to push these boundaries. It seems like constantly where people are looking at these massive protein complexes now, structural information, all these, there's a woman at Oxford named Carol Robinson. I think she's now, whatever the night version is for women, I'm not sure, but that's what she is. That's how, she's figured out how to modify mass specs and electrospray, these enormous multi-protein complexes, and start picking apart structural information, so you can envision having a protein complex and a drug that you want to look at how it disrupts that complex. There's a lot of things you can start to do with that type of technology, and that's the cutting edge. And so you're talking mega Dalton complexes with non-covalent interactions, really, really high-level stuff. So there is no foreseeable upper limit in terms of what you can actually get into a mass spec, which is phenomenal. You could electrospray a virus in people. Good detection limits of femtomal attomoles. Attomoles are routinely 100 attomoles is routine, and for most things. All right. Yes, please.
How will you optimize the matrix uniformity, like the density versus the laser excitation?
So for MALDI, the question is how do you? Your question is how do you optimize the fluence, or the amount, the intensity, or the rate at which you?
How do you know that your sample is having uniformly distributed, or how it's going to excite uniformly in each different places?
I think the short answer is it probably doesn't, because there are, I mean, everything, of course, when it starts off in the early days, people were trying lots of different parameters to try to see what are the best conditions. So rep rate, how intense, how much wattage you're putting into the surface, again, the ratio of analyte to sample, the type of, so the penetration depth. So I think if I'm answering your question right, the penetration depth, I have these slides on my bigger slide deck for MALDI. And to be honest with you, I cannot remember the actual penetration depth. But I've got SEM images of craters where they've hit, but it's usually been multiple times. But I think it's on the order of nanometers as far as the penetration depth, what actually gets off the surface. But that could be, I'm trying to give you a number, but I don't know if that's complete. So I'm having trouble remembering exactly what those slides said. But a lot of that's been put into now, which you would probably get in a modern MALDI-based system. A lot of those parameters that you used to mess with are really been squeezed out of the system, and they've really probably got the key things that are going to give you data. And also you have to remember that the detection, the analyzer transmission, so the extraction, the transmission, the detector technology, it's all gotten better. And so you don't have to be as dialed in probably as you used to get good signal. I don't know if that answers your question. Probably one for a more detailed offline discussion. Good question. Lots of different classes of molecules certainly can be ionized with electrospray, but the key is ionic. Obviously you need, but the key thing here is that polar anionic, but also molecular weight. That just speaks to this issue of there's no really hard upper end limit to the molecular weight of what you can look at, but you do have to have some charged ionizable groups on. I think the key thing on this, guy, is that you just have, if we're looking at, this is a capillary, okay, where your sample and your solution is passing through to form this droplet. You have an anode, okay, where you're putting a high voltage, let's say 4,500 to 1,500 volts, all right, is a typical range that you might expect depending upon your flow rate of your LC system. You're essentially electrospraying this at atmospheric pressure, it's out in the air, and you have an instrument inlet that is going into the vacuum system, okay, so you've got this, we can talk about this in more detail in just a minute, but it's essentially electrolytic cells, how I used to teach it to chemists and instrumental analysis. In essence, it is like an electrolytic cell where the gap between the emitter tip and the entrance to the mass spec is really like a resistor, all right? As these charged droplets flow through, and you have a current that is generated. That current is flowing based on the flow of the droplets, the charged droplets, which have a very, very high density of charge on them. So these are some of the parameters that I guess this is how different parameters can affect the quality of your electrospray plume. This is just a picture that I pulled off of a website from New Objective, which is a pretty popular manufacturer of really small capillary electrospray tips, which we buy from, a lot of other people buy from. And so it's showing you, if you think about the solution that you're electrospraying in, the ideal solution, if you will, is a typical 50-50 water and acetonitrile, or a 50-50 water and methanol. And if you have a really small amount of water, like this top left corner where you've got 5% acetonitrile at around 1,700 volts, you really don't have much electrospray being generated, okay? Because your droplets have a lot of water, and so you've got a lot of surface tension that's overcoming that voltage that you're trying to put into it to start breaking it apart and generating the electrospray plume. Okay, so what you can then do, though, at the same voltage is to get to ionize, is you can up the ante in terms of the amount of organic. You break up that surface tension on the water. Okay? Or you can come down here and you can really ramp up on your voltage and keep the solution typically high aqueous. This is important because, I've kind of really emphasized this, is that electrospray allows the coupling of LC to a mass spec. An LC, though, you're typically running gradients, okay, where you start off, for example, in a reverse phase system on a very high aqueous or low organic content, and then you move up to high organic. And so your electrospray plume the dynamics can change with time. It's just something to kind of keep in mind. This is another illustration to just kind of give you an idea of the mechanism, and this one is really the ion evaporation model. There's two. There's one's called ion evaporation model, and the other one's called the charge residue model. I'm not interested in that you know exactly what those two differences are, but charge residue, for the most part, can be thought of as really playing a role in the big molecules, the big macromolecular complexes, whereas the ion evaporation model is the one most people think is what's driving most of ionization in electrospray. But there's evidence to suggest that there's probably somewhere in between. We still, to this day, don't obviously know everything that's going on in this highly dynamic process. So if you look at this solvent, okay, with two different analytes, so this has a red analyte and a green analyte, and I'll have them colored here for a reason. If you put a really high voltage on this anode, remember that illustration that I showed you before, so you put 2,000 to 3,000 volts on here, as that thing flows, you end up generating this charge, really excess. If we're in positive ion mode, we have this excess of positive charges that form at the tip, and it forms what's called the Taylor cone. And as that flow continues to move the salt solution through the electrospray emitter, you end up generating these parent droplets. Okay, and these parent droplets include the analyte, certainly the solvent that you're electrospraying in, and then this charge excess that surrounds the surface of the droplet. Right? These charges have no interest in being near each other; they want to get away from each other. Okay, so the first thing that's generated is this charged liquid droplet with your analyte in it. As it moves closer to the inlet of the mass spec, which is on the far right of this slide, evaporation starts to occur. So if we're looking at this, which is a really low flow environment, you don't need as much heat into the system. But in a high flow environment, so like the newer system that we have, you're going up to like 400 microliters a minute, 300 microliters a minute, you're going to need to pump in some heat. Because if you think about what you're doing, as these things lose solvent molecules, the temperature of that droplet goes down. All right, it's taking energy away. It's like what happens when you perspire in the middle of summer and you're sweating. Okay, the idea is that as that perspiration, that sweat evaporates, it takes heat with it, cools you down. If you don't put heat back into that system, you'll basically end up electrospraying ice cubes at the end of the day. So temperature is a factor. As you get closer and closer to the inlet, okay, as evaporation continues to occur, so now we're kind of looking at that bottom figure that's showing you that parent droplet of maybe less than 5 microns. So as solvent begins to evaporate, those charges get closer and closer to one another. Okay, the volume, or the surface area, I should say, of that droplet becomes smaller because the volume of the droplet's going down. So there's less real estate for those positive charges to get away from each other. So they're being forced to get closer and closer and closer together. And when they reach about 80 to 100% of what's called the Rayleigh limit, you end up undergoing fission, and that's when you end up getting this ion evaporation process where droplets will be released from the parent droplet to free up more space for more charges to kind of go, okay, now we're a little bit more relaxed. But then evaporation starts happening on that guy again, then all of a sudden fission occurs, and so it's this progressive process towards the entrance of the mass spec, where eventually the analytes that are at the surface of that droplet escape. They have enough energy to escape the droplet, take charge with it, and then basically get into the mass spec for mass analysis. Now, the colors of the red and the green are meant to show the effect of hydrophobicity. Okay? This is really important when you start talking about carbohydrates, because with carbohydrates, you're talking about something that is very, very hydrophilic. Okay? Carbohydrates heparan sulfate is the most hydrophilic biomolecule known. It's got the most charges. It's incredibly soluble. And so those guys are really tough to ionize if it's competing with something else in the droplet that is more hydrophobic, because that more hydrophobic species will go to the surface over staying in the core of that water organic matrix, if you will. Okay, so there's this interplay between what stays in on average in the center versus what gets to the surface, which is where all your charge is and where it's going to eventually get enough energy to release from that droplet and become a standalone analyte, protein, peptide, carbohydrate with a charge on it. So more hydrophilic things stay in the middle, more hydrophobic things stay on the surface, and they're typically going to get ionized more favorably than the things in the middle. This was shown with, do you have a question?
Yeah, I was actually wondering, so if you change like the organic composition of your mobile phase or something, and you increase it to, like you were showing before, like 70% or something?
Yeah.
So at that point, would you expect your heparin sulfate to actually inject faster because you have like more organic, I guess, more organic character in the mobile phase or something?
Well, so I think that, good question. I haven't thought of it that way. So there's a couple things. One is that it's usually, so if you think about it from a chromatography standpoint, those things that co-elute, those things that elute at the same time, in general, from a reverse phase standpoint, have the same quote-unquote, not hydrophobicity, but they're in the same ballpark. Things that come off at the beginning of the gradient are going to be the most hydrophilic, and those things that come off at the end of your reverse phase gradient are going to be the most hydrophobic. So when you think about ionization, competition for ionization, it's on an LC-MS experiment, it really comes down to what's in there competing and what. So then it gets to not just hydrophobicity, but also chargeable groups. And heparin sulfate has a whole ton of those.
So you would probably have a lot of the same like species in a droplet or something, is what you're saying?
Depends on the sample, yeah. So like if you're looking at something, a biological fluid or cell, you're going to have a lot of things that are going to come out at the same time. And they can be based on hydrophobicity, but it can also be size, just also the 3D rotational, whatever the, I don't remember the name of it, not the rotation of, there's some kind of term for it, but whatever the degree of rotation or something, which is how big the hydrodynamic volume is of a particular molecule. So all those things can play a role, as well as the chemical interaction with the surface versus the, not just hydrophobic, but there's also some producing chemical interactions, ion interactions, that kind of thing. So to think about this, and I don't have a good answer for you. So the answer is I don't have a good answer for you, but it's something I need to think about. So it's been a while since I had my surface science class in color and stuff that I think about. So moving forward, just to keep going, don't want to just, so I'll just throw up my hands and say I don't know the answer. I'll have to find out. But moving forward though, this is just kind of showing you some of the some early work that Finn did that really kind of, I don't think these little purple arrows made it into your copies. So those are right here. But what he did in this very simple experiment at the very beginning stages in 1993 is he took 3 different alkylamines. Okay, so he has an amine group in the middle, and then he has alkyl groups, quaternary amines, some of which, so the first one is a methyl, the next one is a butyl, so 4, and then the next one is heptyl, or 7. So he's increasing the number of carbons, or carbon length chain on the quaternary amines. And he's mixing these 3 things in a 1:1:1 molar ratio. And he's electrospraying them at the same time. And so all 3 have equal molar mixture, same solution. And what's happening, though, is that the heptyl dominates. And it's all based on the hydrophobicity of that that analyte versus the butyl, which is in the middle, and then the methyl, which is at the bottom. And so I've shown you here some of the red arrows. This is an expression that he had in this paper with all the things that affect or had a role, play a role in the electrospray response, the ion flux. And so the red ones are the kinds of things that the radius of the droplet, so flow rate. I'm not going to get into that, but that's a huge one. Distance of the ions, how far away from the emitter to the entrance to the mass spec. Those are things you can control. Temperature, I can control the temperature, my countercurrent gas, other things. But those are the three things I can control on my mass spec and my LC and everything else. The things in purple, the hydrophobicity or the nonpolar surface area, if you will, those are the things that I can control post or pre, I should say, in the sample preparation. I can tag things, and because we're going to talk about tagging in the glycosylation section, I really wanted to emphasize this point, especially because when you tag something and you change its nonpolar surface area, you add hydrophobicity, all of a sudden you've changed its ionization efficiency. So you guys in Dr. Desai's group will see this. I don't know if you're aware of this or not, but if you guys use a lot of the ion pairing agents, hexylamines, octylamines, things of that sort, you will see very clearly, if you change the length of the chain on those ion pairing agents, you will get a bump in ionization efficiency, all things being equal. And that's the same effect. Okay? This is just some of the early work with proteins that shows you can use iodoacetamides. This is an old molecule that we've worked with. I don't want to really kind of run out of time. But you can derive. So this is how we kind of got started doing derivatization in an old lab with proteins and peptides. If you put something, same kind of concept, something with higher and higher amounts of carbon or carbon length on it, you're going to see an advantage in terms of electrospray response. So in this case, for a 1-to-1-to-1 ratio, you're seeing something like a 16-fold increase, okay, from the regular derivatized up to decal. You can take it even a step further to some of the neuropeptides and other type of coagulation inhibitors, things you can just get small little peptides. Put in these types of tags, you can see up to a 100-fold or 1,000-fold increase in ionization efficiency. So these derivatization steps can provide huge analytical advantages, and that's why we spend time thinking about how to balance the amount of how much time do we want to spend derivatizing or developing a derivatization strategy for aniline glycan or heparin sulfate, and how does that impact our chromatography, how does it impact our ionization, and how does it impact our ability to detect and quantify things. So derivatization is a big part of what we'll end up doing here with this new system in glycosciences. And this is just the data that shows it for peptides, but it all plays a role in carbohydrates. These are just some of the nonpolar surface area numbers for some tags that we've kind of developed early on. This is showing you some of the responses. Just trying to get through some things pretty quickly because we still have a good bit to get to. This is just showing you some hillet data for some N-glycans for LCMS. So this is hillet, not reverse phase. So the elution order changes, but you can see that for things that have a higher nonpolar surface area, so this phenyl GPN hydrazide tag versus this GP hydrazide tag, which is a 108. It's also got a precharged cation. If you look at these guys, so if you take a 1-to-1 to 1-to-1 mixture with this glycan right here, see if this guy's away. If you derivatize this where this R group reaches all these different things, you can see that in hillet, the thing that is the most hydrophobic comes out first, okay? And that gives you the highest electrospray response versus those things which have, there's no derivatization, gives you basically very low signal. So this is something that's kind of really driving home the point that derivatization can help you in terms of abundance and electrospray, as well as it can also tailor your chromatography. So these are some key points, which I'm not going to belabor to keep going. But the bottom one, which I did not add, I added, I guess, later, is this issue of you can tailor the electrospray response to chemical derivatization. And that's a really important factor when you're talking about these really hydrophilic carbohydrate molecules. And so there's a whole host of literature out there just focused on that alone. So now let's quickly move into mass analyzers. I'm going to kind of pick up the pace a little bit because I'm probably chatting a little bit too much about ionization, which is important. And I may blow through, or I might actually skip, I think, the time of flight and go straight to the Orbitrap for the sake of time, because I'm kind of running over right now. So there's a lot of mass analyzers, and when I say mass analyzers discussed in this lecture, it's another lecture which we just kind of hack this guy out of. But all of these play a role. That's the key point I want to make about this slide is that quadrupoles, 2D traps, TOFs, and Orbitraps, you can mix and match these guys in lots of different ways to get the MSMS or MS to the N capabilities. So you can put a lot of these guys, you can put all of them together, but there's only certain configurations that you'll find because of patents and companies sell one version versus the other and that kind of thing. But for all intents and purposes, you can pretty much stitch all of these things together in one way, shape, or form. So let's just bump down to the Orbitrap. But if you look at like the triple quads, if you want to do targeted PK/PD studies, you're going to use a triple quad. They're ubiquitous. They're everywhere. It's great for quantitation, but it's terrible for, you know, identification and other types of things. Q-traps, linear ion trap or LTQ Orbitraps, quadrupole time of flights or Q-TOFs. Those guys are very, very common. Quadrupole Orbitraps. We have one of those in my lab in the other building. The Q-Exactives. There are TOF-TOFs. There's now only one company. Bruker's the only company that makes these guys, but they actually give you a higher energy HCD fragmentation. So for like multi-imaging techniques, they're really, really nice. And then there's also the instrument we just got, this Fusion Lumos. There's a quadrupole, linear ion trap, and Orbitrap. There's 3 different mass analyzers in this system with at least 4 different ways to do MSMS. So, you know, whatever, pick your poison day. It's like every day of the week you can do a completely different type of experiment on that system in terms of the different combinations that you have. So, as I said, let's kind of skip through the TOF stuff and go to the Orbitrap. These are just some of the developmental milestones and when things got developed that you can look through. TOFs still are still very relevant. They're very fast. They have very high rep rates. Okay, but they don't have the analytical figures of merit as an Orbitrap does. So when you start talking about mass accuracy, how accurately can I define the mass of my target biomolecule or its fragments? An Orbitrap is far superior than a TOF. Okay, resolving power, mass accuracy. But what a TOF has is it has very high rep rates. You can get lots and lots of, you can do lots and lots of experiments in a very small unit of time that you maybe can't do as well in an Orbitrap. Okay, Orbitrap's a little slower, right? Why is that important? When you have really complex mixtures and you've got a sample that you put on the front end of your LC and you're running a gradient, and all these things are being ionized at once, and you've got to measure their intact mass and then get fragmentation on all these different things, you've got to be fast, and you've got to be very efficient. And it's really challenging to do that if you don't have a really fast instrument. Okay, so you're going to miss things if you don't have a fast system. So TOFs have traditionally been very, very good at that, although the Orbis are picking up. So again, let's just leave it at that, and I won't go into any more detail just for speed standpoint. So from an Orbitrap, so the advantage is you can get better than 3 parts per million mass accuracy, so like something like 0.001 Dalton mass accuracy, depending upon the mass or mass to charge of your analyte. Okay, so very, very accurate measurement of mass. But the tighter you can restrict the mass or tell somebody with a higher degree of confidence that this is the mass to charge, i.e. the molecular weight of your protein or your carbohydrate, plus or minus some mass accuracy, the more confident you can be that it's this or only these other possible few other things. The bigger that window, so the poorer your mass accuracy, the more things can fit in that mass to charge space, right? And so mass accuracy really helps you deal with that. The resolving power, you can't get mass accuracy if you can't resolve these peaks really, really well. So the lower field Orbis are like 140, all the way up to the ultra high field, which you can get now to 1 million, which is just phenomenal. It used to be you have to use like FTICR or something like that, which you don't have to do anymore. Duty cycles 10 to 30, that's probably now with the fusions, it's up around 50, depending upon what you want to do. They are compatible with electrospray and MALDI, but for the most part, they are dominated by electrospray. good limits of detection, lots of tandem configurations, as I mentioned. The disadvantages are the linear dynamic range is not as great as, say, like a triple quad, which has like 5 orders of magnitude. You need really, really low vacuum, all right, so 10 to the minus 11, 10 to the minus 12 torr, really important, and the cost can be quite exorbitant. So what does this look like? I chose not to use the animation because I didn't know how well it would work on the two-place over the go-to meeting, but there is a thing called Planet Orbitrap that has all kinds of different animations. I probably showed it, I think, in the course earlier this semester or last semester, whenever I taught that. It was just a mass spec heavy discussion. It's a really great place to go for animations to get an idea of how these instruments are working and what kind of different experiments you can do. But for the most part, in Orbitrap, the reason it's slower than a TOF is that a TOF pretty much is an event where you have an ion packet that reaches a detector and it slams into that detector and it generates a certain amount of current. And Orbitrap doesn't work that way. Orbitrap takes the ion packet and it gets caught in this C-trap, and the C-trap kind of focuses it a lot like a lens or something does in a microscope. It focuses light. This guy focuses the charge, or all the ions. So you get these guys trapped in the C-trap, and they focus it into a packet, which then moves into this Orbitrap. And this is the picture in the top right corner of it. It's about the size of a $50. Okay, it's not very big at all. All right. So the ions come in, and they see this center electrode, which is sitting at a negative. So if I'm looking at positive ions, they come really flying through here, and it's off axis. So as soon as they come into the Orbitrap, they're a little off axis. If I'm looking down at that center Z electrode, and as soon as they see that, as soon as they get to a certain distance, they feel that really high negative 15 kilovolt voltage. And as soon as that positive ion packet feels that negative ion pull, it starts to do a turn, and it starts to loop around, and then that constant loop starts to form this motion, okay, that theta, or delta, I guess that's a delta, around this z-axis, okay? And so as soon as you generate this motion, you start to drop that voltage because you don't want to create this situation where the ion packet gets closer and closer and then they run into the electrode, okay? All you want is for them to make that turn, get going into an Orbitrap motion, and then you want to kind of drop that voltage and then let them basically oscillate along that z-axis. So if we look at that more carefully, there's 3 different forms of motion. The key is this omega z, which is the axial frequency. And so as I said before, in a TOF type system, you've got basically the thing that defines the resolving power and ultimate mass accuracy, not ultimately, but it impacts it, is the kinetic energy distribution, or the kinetic energy needs to be really, really tight so that you don't have a lot of distribution, or everything's pretty much sitting at the same kinetic energy, and you're measuring the differences in the time of flight. Once they reach the detector, they smack into it, it's destroyed. It's a destructive technique. The Orbitrap, however, comes in, and it starts to rotate, okay? And so you've got that minus Z and plus Z, which are basically a detector plate, if you will. And these guys measure what's called the image current. So this red packet moves back and forth, okay, very, very fast, okay, to generate this sinusoidal curve, where you have the image current is generated for, or the frequency. So you're measuring the ion's frequency. Everything that has a distinct mass to charge has its own frequency. So you're not measuring mass to charge, you're measuring frequency. That's shown in this expression here, where you have the axial frequency is equal to the square root of some constant k divided by mass to charge. Okay? So the k is like a lot bigger, obviously a lot more things go into that. But in essence, the frequency, the higher the frequency, the more measurements you have per unit time. Yes, sir?
So the frequency being measured is the going back.
Exactly. Exactly. It's going back and forth. Okay, you go to Planet Orbitrap and you look at the products. Maybe I'll try it here in just a minute. I'm not sure. I just don't want to foul up the guys on the Wisconsin side. But But if you go to Thermo Fisher Planet Orbitrap and you look at different Orbitrap types, you can go in there and they have these really nice animations that describe things from electrospray ionization all the way through, and they'll show some of these same types of pictures but animated that'll be a lot more probably informative, or it'll make more sense once you see that as well. But you're measuring exactly this oscillation frequency along the center electrode. Okay, it's probably, you know, on a given like say in a half a second to a second, it's probably traveling a couple hundred miles. Okay, so in 1 second or half a second, depending upon the actual, if it's a high field Orbitrap or a regular one, let's just say it's traveling a couple hundred miles in that half a second to a second. So you're talking about a ton of distance that's being traveled in a very short amount of time. So, as I said with TOF, it's destructive. It's not with Orbitrap. You have a lot of flexibility with the Orbitrap. The longer you listen to that ion packet within reason, the higher the frequency, or the higher the resolving power. If I cut off the time that I listen to that ion packet, I'm going to get lower resolving power. All right? The longer I listen, the better things get to a certain extent until the things start going out of phase and bad things start to happen, which you're not going to get into. But between 0 and 1 second, all right, that's essentially what you're dealing with. If you've got a 10-second peak that's coming off of your LC system, and you've got 50 things that are coming off at any given time, you've got to do at least 50 MS events before that peak comes off, and there's all these other peaks coming and going at the same time. So a second is a lifetime on an LC time scale. But when you think about the complexity of the spectrum that you're seeing. And the last thing I'll mention is that the mass to charge, the multiple charging aspect, okay? The lower the M over Z, the higher the frequency. The lower the frequency, or the lower the M over Z, or the higher the frequency, the better things get on Orbitrap, okay? Because the frequency, the higher that frequency, the more oscillations you get per unit time. So when you have something, let's say a 10,000 molecular weight protein or heparin sulfate chain, all right? You put one charge on it, and it has a certain frequency, a mass to charge of 10,000. If I put 10 charges on that heparin sulfate and I fold it from 10,000 M over Z to 1,000 M over Z, I drastically changed its frequency. And in the same amount of time, I'll get much better analytical performance out of that 10 plus charge state versus, or 10 minus charge state versus the 1 minus, okay? So that's why multiple charging, electrospray is so advantageous in FT, Fourier transform-based systems like the Orbitrap. That's why you don't see as much of the multi, because it's only singly charged things. Right? Yes.
So the frequency is not depending upon the mass, but it depends upon the charge.
Mass to charge. Yes. Mass to charge.
Both.
Yeah.
Yes. So typically in an Orbitrap, you're not going to be looking much above, let's just say, 3,000 to 5,000. Unless you're looking at like the intact protein stuff, you can really extend that out, and they you can buy instruments that are kind of set up to do that, where the optics and other things are set up to really accommodate these larger. So it's not impossible. It's just that you're on a shotgun proteomics, or the kinds of things that we do with glycans or glycopeptides. You're really living in that 2,500 mass to charge to, say, down to 300 or 400 mass to charge. And you really don't deviate too much from that window, unless you need to. And you do it, if you have to, you know that the higher you go, the less your analytical figures in there are going to become. Okay? This is just showing you some this. Oops, wrong direction. So I think for separating, if you miss a few or you're off by 95% or 90% efficiency, it gets to be really challenging with the number of different things that are in there that can throw a lot of complexity your way that you're really trying to avoid. But then, of course, all of these can then be moved to the different types of approaches, and you can see there's a setpack C18 to purify or do salt for methylation. There's also these digestions, if you will, to kind of chew up different things that might be on there, salidases, things of that sort. And that is for the N-glycans, which of course is the P and GAZF, which we'll talk about. And then there's also the O-links. This goes from a think a glycoprotein as well as then a glycopeptide. So again, not to belabor it, there's a little bit of redundancy here versus the other one, but a lot of steps have been condensed now with this new technology with the Lumos, where now you don't have to release the N-linked glycan to get the identification of the peptide and the carbohydrate, okay? So that is a huge transformation that's starting to occur. So all these different workflows, and in every step, there is the opportunity to introduce, pre-analytical variability, introduce artifacts, things that are going into the carbohydrates that are not real. And so all of it collectively is getting shrunk constantly. And the less you have to do with your sample, the happier you end up being. So let's look at some of these release chemistries which are commonly used. This is a hyperlink I won't go to, but this is off of NEB, which is a nice website to kind of show this animation and kind of pulls everything together. Perhaps if you're a little bit not super comfortable with my delivery, the animations always help. So for N-linked, again, you have these guys are linked to proteins with a known motif. This is asparagine, linked to any amino acid, or this X, followed by a serine or threonine. So the asparagine is on the N-terminal side, moving towards the C-terminal side with X and serine or threonine. This is a conserved core, so when you are, you can predict it. Albert, you can say this is a predicted site. You already kind of have an idea ahead of time that this is a good candidate region in the protein that already probably has N-link glycosylation, or could very likely have it. All right. There is a conserved core, okay, structure, which I'm showing you here at the bottom with these two Glycomics bound to the asparagine up to this branch structure with 3 monosaccharide units, and then off to the different forms it can have. And then again, as I go back to the Cummings review, there's somewhere around 3,000 relevant structures that are believed to exist in humans. And that includes O-links, so it's probably less. These are just some of the general classification that you can have these high mannose. So this green again is the mannose groups. Complex, where you have galanax, or as galactose, sialic acid groups, which are really, you see a lot more sialic acids in things like cancer, tumor tissues and things of that sort. So I remember it's been at least 10 years ago now, looking at going to presentations and hearing that people argue, mass spec people argue, over the best way to look for sialic acids. And the biggest, one of the biggest arguments that you would hear people have is that you don't use MALDI because it pops off the sialic acids. And people, big groups would get into all these kind of arguments. And I not having any direct experience with it or not, I can't say one way or another. But I've mentioned this before, and I'll just mention it again. This is one of the reasons that, I guess, perhaps MALDI is not as widely used for these types of things because of the fear that it's, although it's soft, it's not as soft as electrospray, and the worry that you're going to miss some of these things. Okay, so there are 2 main enzymes that you can use to release the N-glycans from the protein backbone. The one is this endo-H, which releases it between 2 GalNAcs, and one is PNGase-F, which releases the GalNAc from the GalNAc and the asparagine residue. Again, here's that motif that I mentioned to you before: asparagine, X, and then serine or threonine. And if we go to the next one, yes, sir.
So these enzymes, what are they recognizing? Are they recognizing the motif or the GalNAc?
I do not know the answer to that. I'm not sure how they recognize. I do know, or I think this is true. If you have a fucosylation on this first GalNAc, that can inhibit. That's one of the things. But I think fucosylation is primarily seen in plants as opposed to mammalian systems. So it's not as of an issue, I think, in mammalian or proteomes, glycoproteomes, if you will. But as far as what it's recognizing, that's a good question. I do not know the answer to that.
Because if it were recognizing a GalNAc, or like 2 GalNAc residues together, then, I mean, a lot of glycans might have those.
That's true.
So would they also just break that segment off as well?
Well, so you know what? The question will make me go back in the literature and figure it out. I actually don't know the answer to it. I'm not even sure actually the PNGSF, or Indo-H. I'm not sure if that's bacterial or if it's what's the history behind that, like with as opposed to like trypsin. I can tell you why we use trypsin and where it comes from in proteomics, but it's a good question. I don't know the answer to it. I don't know if it recognizes the protein.
Maybe look for 1,4 linkage. The 1,4 linkage, the Indo-H. So if you see the two GalNAcs, the linkage is 1,4. If you target the 1,4 specific.
It could be, but the 1,4, I guess what he's saying is like if you have a 1,4, I guess you wouldn't have, that would, in theory, the GalNAc, because you have two, don't you have two Glycomics in a heparin sulfate, the 4 core? Did you have a xylose, two Glycomics? No, one's at 1,4 and one's a, I don't know. I don't want to just lead anybody down to some stupid
Heparin is 1,4, 1,4, 1,4, 1,4.
Yeah, just 1, 4 throughout.
Contractin is 1,4, 1,3, 1,4, 1,3.
Yeah, it's a different linkage in the core
Yes.
for heparin sulfate than it is for this. But to his point, I don't know where else you might find the same motif or sequence, the carbohydrate sequence, that could give you trouble. And so does it just, is that a unique set of sequences that it only recognizes, or does it have to also have the protein involved as well? I don't know the answer to that.
So what's like the benefit of using those two enzymes, like cleaving on one side versus cleaving?
Well, like, so one thing you could do is take Indo-H and use it and leave the residue. So if we go to this next one, okay, let me try to answer it here. So I don't really have an answer for you exactly, other than it could be cost. So, for example, in proteomics, there's lysine and there's trypsin. Okay, lysine cleaves the C-terminal side of lysines exclusively, and a lot of people use it. Trypsin does arginine and lysines. And people, you're like, why the heck wouldn't we use lysine? That doesn't make any sense. It's because lysine is like it's like 10 times more expensive at least. I mean, it's a cost issue. And I don't know why it's that expensive. Somebody asked me in the proteomics course, and I don't know why. At this point, you think they could express it. But why would you use one enzyme versus the other if you're doing an experiment where, in the old days, for example, you're trying to find site localization of in-link glycosylation, and you want to know, does my protein or where in my proteome is there in-link glycosylation occurring? If you do a PNGASF treatment, okay, you end up taking asparagine to an aspartic acid, so you end up increasing it by a Dalton. So when I go into take my shotgun proteomics data, after I've released all the PNGASF, or all the in-link glycans with PNGASF, I'm going to put in a variable modification for asparagine to aspartic acid. It's a if this maybe is a possibility. Okay, it's a 1 Dalton shift. But there's the other aspect which you could do the Indo-H and leave a little bit more of a monosaccharide residue, which may give you a more distinct signature, if you will, that, oh yeah, this was definitely in-link glycosylate. But so I don't know the answer, but I want to say, what's the cost of Indo-H versus PNGASF? What's the efficiency? Are there exceptions? Does Indo-H have exceptions where it doesn't cleave high mannose as well as hybrid? Versus, there's these kinds of biases that can be present that, I just don't know off the top of my head that are something to consider for sure. Okay? But this was the standard workflow. You basically hit with PNGASF, release it. Now you have this 1 Dalton increase, and you're looking for that in your data set to see if, oh yeah, there's an aspartic acid that should be an asparagine, and it's in the motif, treated with PNGASF, therefore it was in link glycosylate. So there's a lot of papers that focused on that 10, 15 years ago or so. So this is some data, just some old data off of an LTQFT, at least 10 years ago, where we treated human plasma with PNGASF. We didn't do any derivatization, no reducing and tagging. And so you can see this is the these are the peaks of different types of glycans, and I'm highlighting this one right here in this burgundy, this gray box. So if I'm looking at the intact mass spectrum of that peak, that's shown here in the middle with this red dot, the monoisotopic peak. All right, so you guys in the mass spec course, you guys are, kind of hit you guys over the head with the monoisotopic mass. How do you calculate it? This is the peak, the most isotopically pure peak that you use for identification, or at least for the initial identification. So you've got this glycan has a 2+ charge state. This is its measured mass to charge, and essentially this structure would give rise to this mass measurement accuracy about 1.55 parts per million. If we now isolate just this monoisotopic peak, so just the red dot peak, not the other M1, M2, and 3 peaks, when we fragment it, you can see the different fragment ions that are associated with these red lines showing you the different fragments that can occur. And from this standpoint, it's probably like, so this sialic acid could be here or here. So that's some of the issues that could show you really important biological information that I don't believe that this information right now could give you. Okay? So these could be stereoisomeric, biologically significant stereoisomers and things of that sort. They're tricky to tease out. Okay? But that's essentially the 3 elements, the 3 data types that you're going to encounter in this particular type of an experiment. The chromatography, with all these different peaks, all these different glycans that you don't really know what they are. At this point, you just know there's signal there, and they have mass to charge, multiply charged mass species that look like they're probably carbohydrates. You get their intact masses. So I've got the second piece of information, and now I'm going to start fragmenting. And this is just a standard MSMS. This was done in the linear ion trap, the LTQ. On this new system with the Lumos, you could do, high energy collision-induced dissociation. You could do MS to the N, multiple levels. So each one of these fragments, for example, you could, after you get this fragmentation spectrum, you could then go in and isolate each one of these peaks and do additional levels of MSMS. Fragment it, fragment this one, fragment this one. So there's a lot of different levels of complexity that you can get into with the newer instrumentation you can take. These 3 pieces are the key foundation for what you're going to use for identifying unknown glycans, particularly those that are not template. We don't have anything to match this up against unless we have like a spectral database or we're using a piece of software like SimGlycan, which could really help us in some ways. We're just getting started with that. Okay? So O-link glycans, these guys are much more challenging, okay, to study. And the reason is because they we functionalize serines and threonines, but there is no conserved amino acid motif. So like with N-linked, you've got asparagine, X serine, or threonine. These guys don't have that motif that you can use to predict whether or not this would or would not be O-linked glycosylated per se. There's 8 basic cores that can branch off into linear chains and other types of things. And again, that's the numbers of structures are within this 3,000 plus glycan structures as well. That includes also the N-linked glycans based on the Cummings review. You can see that if we're looking again at the N-terminus of the protein all the way through with the serine or threonine all the way to the C-terminus, you've got a GalNAc, okay? And then you have all of the different core structures which can come off of that. So these are the 8 structures from this review article from 2015 from Heather Desaire's group, core 1 through core 8, and just the different variety that you can get into accordingly. There are enzymes that you can use to release them. You just can't, there isn't one that just cleaves off all of them ubiquitously like the N-linked, which has that conserved core, okay? So you kind of have to whittle away at things. You probably have to say, well, if I use this, and these had this particular core, I should see this loss or this, this change versus this set, which I didn't use, or I used a different type of enzyme. So you have to kind of start to whittle away at things. And for the most part, I think these approaches were generally used for situations where you've got an intact, you've got a fairly simplified mixture of an individual protein that you've IP or purified from something, or it's histag, whatever it is, or it's a recombinant protein, something along those lines that you would use to kind of have enough material to do these different types of experiments. So again, from these different structures possible, generic structures versus the antennae with these exoglycosidases, you can get back down to core 1s, core 3s, and start to kind of, through systematic elucidation, and use this glycosidase to kind of clean things off. These are just showing you some of the different, again, the linkages that get broken. But I think that this slide set is obviously going to be changed a good bit as, again, when you start to move into these new technologies, these new instruments that don't require that you do these types of things, they will still have its own place. You never want to just say, oh, we don't need to use this anymore. There'll always be reasons to have to use something, whether it be to validate or to reinforce what you're seeing in the mass spec, or there could be other reasons. And of course, no matter what you think the mass spec can do with some purified protein from Sigma, and you say, oh wow, look, all our problems are solved. It's never ever in my life been that easy. I've always run into different issues. Okay? So these are some of the things for O-link glycans to release for like removal of O-links using beta elimination. I cannot remember why I put these stars on these slides, but and there's no animation, so it's just a little red star just there for it's watching over us as we give our lectures. So with sodium borohydride, in these types of conditions, you can end up releasing things. And now you're left with this reducing ends here from your protein backbone, which can then go on to be reduced. You can also, now let's start talking about the glycosaminoglycans, the linear guys. This is a much different animal than the branch chain, as many people in this room certainly can attest to. So the number of glycosaminoglycans is probably it's a lot smaller. So like with N-linked and O-link glycosylation for the branch chain, more than 50% of the human proteome is pretty much glycosylated with those guys. With things like glycosaminoglycans, that number is much smaller. There's only known, I think, probably 25 proteins that really are known to have these heparan sulfates, like chondroitin sulfate, keratins, et cetera, et cetera. So the family's smaller, or at least known, but I think that I see papers from time to time, they're like the discovery of a new proteoglycosaminoglycan. And so my guess is these things will continue to kind of be expanded when you start looking at lower abundant proteins that come in through the mass spec to find these different modifications. But in general, you start off with your proteoglycosaminoglycan with your serine that's modified with the xylose gals, and I think the linkage is again different. It's not a one. It's not just a 1,4. It changes, and then you start with the GLICNAC glucuronic acid repeat unit, which can then be changed to an hyaluronic acid with different enzymes. And so you're left with this, for heparan sulfate at least, this long linear chain, which can be anywhere from, say, like, average molecular weight's about 30,000 molecular weight, but it's got some polydispersity to it, so some are less, some are smaller, some are larger. So there's an enormous amount of heterogeneity in this chain as it relates to the protein that it's bound to. And then from this standpoint, you have these differential sulfation with all these different enzymes that can or basically add or release sulfates to the backbone, which can impact protein binding, protein interactions, as well as signaling pathways. And so the structural characterization of heparan sulfate, or at least large chunks of it through enzymatic digestion, is a I would say, much larger challenge than are the branch chain guys, just because the polydispersity and heterogeneity that you encounter is going to be much, much greater. So to get after this, of course, you use different types of enzymes, so heparinases 1, 2, and 3 for heparin sulfate to give yourself the disaccharides, which are shown here in the bottom left. There's about 11 or 12, depending if you don't count the 3-O-sulfate, which can be here. Lots of different options. There's, if you start looking at the numbers, again, if you go back to that table from Cummings, I think a 6-mer, I can go back and find out what the number is. Yeah, so if you take this guy, oligosaccharides, peptides, this is the number of carbohydrates. I don't even think this counts into sulfate or into the sulfation, number of sulfation sites, but it gets to be extraordinarily large, just for a 6-mer, not counting different ones. So how do we go about doing this? Well, just like the inland glycans, fortunately, we have these reducing ends, which give us the ability to derivatize. So once we do a digest, this is for heparin sulfates, you chop it up into these disaccharides. These are just different molecular weights, different R2, R6, Y groups, all of the different options in their molecular weights, some of which are isobaric, so like 2H and 3H are isobaric, exactly the same molecular weight, just difference where you put the sulfate groups. If we then go in and look at the different types of derivatization strategies, directed amidation is one of the probably the most common guy, where R can be an amino acid, or excuse me, an aromatic group or something, or we use a lot of the hydrazide tagging, where the R group is a dual benzene. It's almost like a still bean type, where you can basically put stable isotope labeled isotopes into the molecule to do relative quantification, which we'll talk about. So all the data that I'll talk about, I think, from a while back in my lab, involves the hydrazide tagging, but this is also a common one as well. Yeah, so this is 2AB on a halide column for inland glycan, just to kind of give you an idea. And just to show that this is also used for the branch chains under different conditions. Same kind of concept. So I think the point is that for inland glycans, you've got the same reducing end right here with which you can do these derivatizations as you can for the glycosamine glycans. The chemistry that works for one works for the other. In this case, as I said, we're using this hydrazide tag. This is now sold, it's called Inlight. It's sold by Cambridge Isotope Labs under this Inlight name. If you take, for example, for comparative analysis, unfractionated heparin sulfate that you've extracted from a cell surface or from a tissue, okay, you then end up getting it from tissue 1 versus tissue 2, or cell line 1 versus cell line 2. Treatment 1, treatment 2, whatever the experiment is, you have what you hope is something like equivalent numbers of cells, or equivalent numbers of total protein, something that gives you that normalization like you expect in a Western blot for GAP-DH. Do I have the same amount loaded on the lanes? Yes, I do. Same kind of thing. Once you get that, you can then end up doing an extraction or derivatives, an enzymatic degradation, and then it end up reacting again with this free reducing end with this hydrazide tag with either a carbon-12 or carbon-13. So every single carbon in this benzene is either purely carbon-12 or purely carbon-13. So ultimately what you would end up getting is with 6 carbon-13s, that gives you a 6 Dalton shift relative to the naturally labeled. So if I label sample 1 with my light hydrazide versus sample 2 with my heavy, and I mix it together 1 to 1, all right, everything's being equal, and then I run it by mass spec, I now have basically a way to do internal or relative comparative quantification. So this is what it looks like for a disaccharide. These are standards that you could buy, and just showing you where this guy essentially fragmented using the different Costello and Damon nomenclature, X, Y, Z, ABC. You can see here's the tag, different sulfation. So this is for the 1S disaccharide, which is a common one that you would see. If you're measuring this, this is a doubly charged internal standard with a light tag versus a heavy tag. This is at a 1-to-1 ratio So you know that the height of these peaks is roughly 1 to 1, all right? If I then isolate this guy and then do MS-MS at a collision energy of 22, I'm left with, again, all of these different fragment ions. This just shows you how changing the energy of fragmenting these ions can really impact the amount of information that you get, so you have to do a lot of optimization before you kind of dial into what works best. You can see that between 20 and 25, that gives you kind of an apex of a lot of different fragments from this species. And this is the intact, it's basically going down. So with this, I can now do higher molecular weight oligomers, okay? So this is a DP8 that you can buy a standard for. Again, this has all been tagged. So you can actually tag it, mix it together, 1 to 1 from our method development or a theoretical standpoint. So this guy, now that you're seeing a little bit, you're starting to see some overlap with the isotopic distributions. This is a 3- charge state for this DP8. This is the light labeled, and this is the heavy form. Okay, if I end up doing collision at different energies, then one of the challenges and the reasons that you end up having to go to ion pairing agents, right, is that your sulfates start popping off before you actually get any information. And if the sulfates, or the location of the sulfates, which you know is so critical to protein binding, if you can't find or can't say with any confidence where the sulfates are, you've essentially lost all of the biological information in the analysis. And so even with the tagging, which you can get quantitative information, we're still stuck to where when you start to break down this molecule, when you isolate this peak, when you start doing MS-MS, you end up losing all the sulfates. Okay, so each one of these is a sulfate loss, each one of these dots. All right, and as I increase the energy to fragment and break that molecule apart, I end up stripping it completely down to this purple one, which is, I think, it's got like 5 or 6, I can't remember, sulfates left on it, where you can just start to see fragmentation peaks down here in the weeds. And then you move up to a collision energy of 35%, and then you get all kinds of fragments, but it's too late, all right? Not too late, but it's like, it's all for naught, because you've lost all these sulfates in the process. And that's why you end up having to do ion pairing agents, and why we're so happy to have this new instrument, because I don't have to put ion pairing agents on my rototrap because they will kill that system for the rest of its existence. All right. So in heparin sulfate, with some of the challenges, separation, which I haven't even touched upon, but a big ticket item again is sequencing these guys in the mass spec is a huge, huge challenge. And the key is that you're losing all this information, even under soft conditions with electrospray. You lose these sulfates, and that's where all the information lies for us for heparin sulfate. That's just a total ion chromatogram. These are just some extracted ion chromatograms showing you the individual peaks and where they're coming off for retention times. So this is a reverse phase experiment where each one of these things has a unique mass, or these are the same masses. All 3 of these have the same molecular weights. And so when you say, give me just these, the things that are molecular weight plus or minus some mass accuracy, you're getting 3 different species. This guy, you get 3 species. Should only be 2. These guys are singles, and these guys are 3s with some other noise. But again, it's this 1 to 1, this ability to do quantitation, as well as when you do MS-MS, which if you look at the fragmentation, you can start to see where's the things that contain the tag and which things don't. So this 157 peak, that's common to both. That obviously does not have my tag. You know, then you start to see these 6 Dalton shifts. Those tell you that these fragments contain my tag, and that's a really important piece when you are having to go through and essentially or manually interpret all this data. All this data is manual interpreted. It'll drive you to, I mean, I did all of this myself, let's put it that way, all right? It took about 6 months between doing a lot of other stuff. Just sit there and go through, manually extract all the data, interpret the MS-MS, and validate what I think is real and not. And that doesn't account for even what the oligos. So we saw some things that were the DP6s, DP4s, and some DP8s. This guy is showing you a DP6, and this is a DP8. This is kind of giving you a molecular weight distribution of some of the things that we saw. Saw about 78 different things that we think are unique. So again, it's all manual interpretation. This is why carbohydrate analysis and mass spec is such a such a ripe area to be focused on. And I just feel like I'm in the deep end, just trying to keep my head above water. In proteomics, it's like, oh my gosh, there's got databases, I've got Sequest, I can just submit my data, go home, go get some coffee, come back, maybe it's got the data ready. There's all my data nice, all tabulated, identified, ready for my Nature paper to get rejected. Go into something like this, it's a hard grind to get through all the data. There's really there are some academic pieces of software out there that do okay, but nowhere near the ability to go in and really identify them and also detect which things are different. So from our standpoint, tagging allows us to say what's different. That's the first thing. We don't know what it is, but the idea is that when we start labeling sample 1 and sample 2, we mix it together, we want to be able to say, after we normalize, that if we see a difference of less or greater than, say, let's just say a half a fold or a couple folds, there's something unique about that oligomer, that structure of that oligomer, in sample 1 versus sample 2. And I need to know what that is. What is that unique sequence? Because that could be the sequence that's causing the difference in binding to protein that's really important in my system. But we still don't know what it is. And so that ability, though, with the tagging to say what's different is the first step that we're going to employ, if you will. And then once we know something's different, we hope to get some sequence information, some idea of what it is. But in all likelihood, we're going to have to come back to the well multiple times and run multiple runs on the system with different conditions, different MS parameters to actually get at what it actually is. Okay, so covered a lot of ground. Probably didn't need, I don't know, 2 hours. Man, these things are killing me. I'm getting so many of these. I've cut things down to so much to the bone that I don't know what else to get after. So, yes, sir.
What is the information that we can get about the linkages in even the O glycans, N glycans, and their MS, and the glycans, the changes in 1,6, 1,4, 1,3, all of those? How do we determine that from these spectra?
Are we talking about the branched O links?
All of them, yeah.
So I think that there's a couple of different ways you can do it. I think that you can use, so if there's a case where you have something, where you have a modification site and you don't know where it is, you know it's there, but you feel like you want to be able to establish where it is because it can have serious implications. When I mentioned to you about using enzymes, you don't ever want to just say, oh, then the instrument's going to take care of it all. If you go back to some of the rules with enzymes, then you can use those enzymes to your advantage. You can say, I'll treat, so I have something which I think is important. It has a modification, but I'm not entirely sure where that modification is. But I can go to the New England NEB website or something and get an enzyme that could remove that important functional group with some level of structural specificity that could help inform where exactly it is. So I'm not sure what that enzyme might be. You might also use other types of mass spec techniques. Things like ion mobility is really making a real charge these days in mass spec. It's kind of moved away from being very academic technology to now much more mainstream and really, really powerful. But you can think about enzymes, you can think about different separation, you can use a lectin column to do differential enrichment, and there's just a host of things. You can think about chemical derivatization. You can think about capping. If you've got something capped with oxalic acid, you can think about trying to cap the other guys with some other chemical group so that when you rerun that same sample through the mass spec, those more stable units that you put on there could give you some structural information. So I think that you have a very large spectrum of options available that you never want to close your mind to You never want to be of the standpoint of, I'm going to get everything I need out of this mass spec measurement. There's a lot of cool stuff you can do with the enzymatic, chemical, just general intuition. But if you know or don't know how to use the mass spec and how to tweak it, it's the same thing as like you become so narrowly focused on, I'm just going to do this. And if I can't get it, I run into that more times than not. So I don't know exactly how, because it's very problem dependent, but I think that you definitely want to be aware of these different techniques that you can use.
What about the linkages between each saccharide molecule, like between, like in a hexasaccharide, like is it one 4 linkage or is it one 3 linkage?
Right, so heparin sulfate versus, I think it's chondroitin sulfate, they have the different linkages.
Yes.
Keratin, I can't remember the 1,4 to 1,3, but if you use something like chondroitinase versus heparin, so if you want to do site localization of heparin sulfate and chondroitin sulfate, for example, you could selectively, so you might enrich everything with a, some kind of an anion exchange column to enrich all those things that have so many sulfates on it. Get rid of everything, digest up all of the heparin sulfate, chondroitin sulfate, proteoglycosamine and glycans, chop them up with trypsin, and then you can go through and you can say, all right, here's my samples. I'm going to split them. One sample I'm going to treat with chondroitinase, and then I'm going to run that by mass spec. And when I'm left with that residual, so if I go back to this guy, so what I end up doing is I chop everything off with my enzyme, and I'm left with this 4-saccharide residue, all right? And so when I run that by mass spec for shotgun, I want to know what the peptide is, but I also want to look for that residue on that peptide. And if it's there after chondroitinase treatment, I know that was a chondroitin sulfate glycosamine glycan. Then the other way is you treat with heparinase, 1, 2, and 3, and if that's gone, then you say, yep, that was heparin sulfate. So you can use enzymes in that way, and it is used that way. There's a couple papers out, several papers out actually on Perlican, and they've shown how they've kind of gone through and really mapped a lot of these sites using that differential derivatization strategy. But there's a lot of experimental design that goes into thinking about it, because you're not going to run that, even that peptide with that huge heparin sulfate chain, you're not going to run that on mass spec and get. It's just going to be it's going to be a big mess, in my experience. And in someone else's hands, it could be. So. All right. Do we have any more questions?
That's it.
All right. So you guys do you guys do a quiz or exam on these things at all?
For the second section, we don't have an exam, we have a presentation.
All right. Sounds good. Because I was Wisconsin and maybe New York.
Thank you.
Thank you.